Hello everybody, this is Nanduram and we are from Sonu Academy. Today we are going to explain about cell division of mitosis. Mitosis. Mitosis is a stomatic or vegetative cell division and the daughter cell resembles the mother cell in every aspect. Organs formed by this type of divisions such as liver, kidney, brain, skin, etc. They are called vegetative organs because they do not participate directly in reproduction. Growth of any organ system in plant or animal is due to the increase in cell number by mitosis. Cells from young and tender organs such as shoot, tips, bud, root tips, tissues of young animals that divided more rapidly than the cells from old and mature organs. This is the reason for a greater rate of growth in younger organs. Before the cell division occurs, various changes are seen in the cell. The major changes is the doubling of the chromosome of numbers. Only after this period completed, the cell divided so that the daughter cell will have the same number of chromosomes as the mother cell. This is essential. You have already learned that chromosomes carry the genes. Genes carry the genetic information and are responsible for the inheritance of parental features. They also regulate all the functions of the cell by directing and controlling all the proteins in the cell. Before cell division begins, the non-dividing or resting cells are said to be in interphase. This phase in the life of cell is called interphase. Chromosomes in the nucleus of interphase cells are in highly decondensed state and forms a diffuse network of filaments called chromatin. Most of the cells spend in a considerable portion of their life in interphase. Mitosis is of a brief duration and once the division is completed, both the daughter cells usually they enter into interphase. Once mitosis is intended, it proceeds without any interpretation. Still the cell divides is completely and the daughter cell has formed. Based on the changes occurs, there are 5 stages have been recognized in mitosis. They are Prophase, Metaphase, Anaphase, Telophase and Cytokinesis. Prophase. This is the first stage of cell division. Before prophase starts, the chromosomes are in decondensed state and appears as a diffused network of filaments. During prophase, the chromosomes start condensing. As a result, the shape of chromosomes becomes definite and distinct. Each chromosome has 2 arms are called chromatids. The 2 chromatids are joined together by a centromere. While these are changes occurring, the nucleus and the rest of the structure in the nucleus disappear gradually. Meanwhile, the centrioles move to the opposite side of nucleus. In the plant cell, where the centrioles are usually absent, then they appear at the stage of cell division. From each centriole, fine thread-like fiber appears. These fibers are called spindle fibers. A centriole with spindle fiber is called an aster. The spindle fiber grows towards the center of the cell. Metaphase. Chromosomes now move to the center of the cell and arrange themselves in a row. This looks like a plate and is called equatorial plate. Some of the spindle fibers are attached to the centromere of each chromosome. Rest of the fibers grow towards the opposite pole and join the centrioles on the opposite side. Each chromosome is attached to fibers from the 2 opposite poles. Anaphase. At this spindle fibers pull 2 chromosomes to opposite ends. As a result, the centromere splits and are pulled apart along with chromatids. As a result, the half of original number of chromosomes move to each of the 2 opposite poles. Telophase. By the beginning of this stage, the chromosomes completely movement and will be at the end of each cell. Nuclear membranes, nucleus and other nucleus structure are reappear. Chromosomes begin decondensation process. By the end of telophase, the shell will be having 2 nuclei, one on each side. Spindle fibers are now disappear. Cytokinesis. A new membrane is formed at the equatorial plate during this phase. This divides the cell into 2 daughter cells. Thus at the end of mitosis, 2 daughter cells which are exactly similar to the mother cell are formed. These first four stages of a cell division involves changes in nucleus and division of nuclear components equally between daughter cells. Hence, these four phases of prophase, metaphase, anaphase and telophase of cell division are together called karyokinesis, means division of nucleus. The last stage is called cytokinesis. Cytoplasm is only the component undergoing division in this stage of mitosis. Points to be remembered in this lesson are: mitosis is a stomatic cell. Doubling of the number of chromosomes is the major event before the cell division. Interphase is the period when the cells grow in size and performs the physiological functions. Chromatin is a condensed state in the nucleus of an interphase cell. During prophase, centrioles move to the opposite poles of the cells and spindle fibers formed from the centrioles. During metaphase, chromosomes move to center of the cell and form equatorial plate. Spindle fibers grow in a length and attach themselves to the centriole of each chromosome. During anaphase, the chromosomes are pulled apart and each half moves to the opposite side of the cell. During telophase, the nuclear member appears around the chromosome and chromosomes decondense into chromatin. During cytokinesis, a member or a cell, a wall appears in middle of the cells and divided the cytoplasm of the cell into two. With this, the cell division is completed and the two daughter cells are formed. During mitosis, karyokinesis occurs before cytokinesis. Thank you. This is Nanduram and we are from Sonu Academy.